Hello and good evening, everyone. So today we'll be talking about a topic that everyone likes and loves, or not, more of a topic of interest. So we'll be talking about plastics and especially a type called polyethylene terephthalate or PET. So we'll be talking about the biodegradation of PET under our hero, the bacteria Ideonella sakaiensis, which is hard to pronounce also. I will be presenting along with my colleague Anirban. First of all, a quick overview of our table of contents. Section 1 will introduce the PET biodegradation. Section 2 will introduce Ideonella sakaiensis as a PET degrading microorganism. 3 will move on to the enzymatic mechanisms in PET biodegradation. 4 will present some case studies on how PET was enhanced. And in the end, we'll provide a conclusion and remarks. So now we'll begin with introducing the PET and its biodegradation. So let's begin with the PET overview. So PET is a widely used synthetic polymer with significant environmental implications due to its accumulation in landfills, water bodies, and ecosystems. Basically, it's everywhere. And I don't know if you guys saw, but there are islands of plastics also in the ocean. Second, for its environmental threats, improper disposal of PET waste poses a threat on the wildlife and contributes to huge amounts of pollution. This emphasizes the urgent need for PET pollution mitigation and sustainable solutions. So the significance of PET biodegradation, why are we doing this? Why is it important? What's the big deal? So biodegradation offers a promising solution to PET pollution by harnessing microbial degradation processes to break down the PET into non-toxic byproducts. Furthermore, the beauty in it is that there is a recycling potential or recycling mechanism. Enzymatic PET degradation do not only reduce the environmental burden, but also offers the potential to recycle the PET into valuable resources. So after degradation, the monomers that we have can be recycled into other products. To the left, you can see a paper also by Ali et al., where they talk about the mechanism, challenges, and future prospects. Okay. So now we'll be moving to section 2, which is our hero, the bacteria that we love, or at least for this presentation, Ideonella sakaiensis, and yes, the N is silent. We'll be talking about this bacteria as well as talking about the enzymes responsible for PET degradation. This bacteria was recently discovered to degrade PET actually, which was in 2016 in Japan. It's a negative gram bacteria, meaning it has a thin peptidoglycan wall. It's also motile, where it has a flagella, and it's rod-shaped. So it secretes enzymes called PETase and amylase, which are crucial for PET degradation, and actually this bacteria uses the PET as a source for carbon and energy. Okay, so now we're moving more into the enzymes of our beloved bacteria. So this bacteria was considered a breakthrough in the field of environmental microbiology and plastic pollution remediation. As we said before, it focuses on 2 enzymes. Take it as one hand doesn't clap by itself. And I guess now you're trying to clap with one hand. Anyways, so it focuses on the PETase and the metase, which work together to break down the PET polymers. The first enzyme is usually the PETase, which catalyzes the initial breakdown of the PET into monomers. And then after that, the metase takes its role and starts degrading those intermediate products, enabling the microorganism to assimilate the PET-derived carbon and energy sources. Okay guys, now for section 3, we're moving more into the enzymatic mechanisms in PET degradation. We're going more into the details of the structure of both enzymes. So please bear with us. So moving to the first step of the degradation, PETase is the first enzyme used. It acts as a catalyst in breaking down PET polymers into other monomers, which are called monohydroxyl ethyl terephthalate, MHET. In abbreviation. So for the structural insights, it belongs to the class of enzymes known as hydrolases, specifically esterases, which catalyzes the hydrolysis of ester bonds present in the PET polymers. Structurally, PTase is composed of amino acid residues arranged in a specific 3D configuration, forming the active site responsible to binding and catalyzing the hydrolysis of PET molecules. As we can see on the top left, we can see that the substrate will enter the binding cleft, and under it we have the PTase molecule. Furthermore, the enzyme exhibits a classic alpha-beta hydrolase shape, including a 7 alpha helices surrounding the beta core sheet that's made up of 9 mixed beta strands. In the second image, we can see the PET entering the substrate and the PTase acting on it. And in this case, the PTase's active site contains a conserved catalytic triplet consisting of aspartate, histidine, and serine residues, which are the amino acids that facilitate the hydrolysis of ester bonds in the PET. And downwards on the bottom left, we can see that the PET is being degraded and it's almost leaving the substrate site. In the second step of degradation, MHET is formed and metase comes in to degrade this monomer into terephthalic acid and ethylene glycol for the energy and food product for the microorganism. The metase also belongs to the hydrolase class of enzymes and acts as an esterase, facilitating the cleavage of the ester bonds. It also works synergetically with the PTase in the degradation of the PET as a whole process. Okay guys, we're almost at the end of the road. Now for section 4, we'll be talking about how to enhance PET degradation efficiency. We will show some case studies, and who knows, maybe we can create one in the future. So we actually found a couple of case studies we want to share with you, and it's always to see those techniques being used to increase efficiency. The first was the development of a mutant or semi-mutant enzymes with enhanced PET degrading activity. The other was the use of a whole cell biocatalyst to achieve higher degradation rates, whereas the third was the modification in the structure or activity of the enzymes, PTase or metase, to improve their catalytic efficiency. All those advancements paved the way for more efficient and sustainable PET degradation methods, contributing to the development of biotechnological solutions for plastic management and environmental remediation. So yes, as you can see, this is our final section, and thank you for staying with us. This section will deal with the conclusion, future recommendations, and challenges. As a conclusion, there is significant potential for the commercialization of PET degrading enzymes for the use in plastic waste management and recycling industries. Enzyme-based biodegradation offers a sustainable alternative to traditional waste management methods and aligns with the growing demand for environmentally friendly solutions. However, there is always the however part. We might face some challenges that include scalability, cost-effectiveness, and regulatory and logistical considerations. For future recommendations, future research directions may focus on optimizing enzyme performance, such as the case studies that we talked about, exploring novel biodegradation pathways, checking other bacteria, fungus, microalgae, and monitoring evolution of the bacteria or microorganisms, as well as investigating the environmental impacts of enzyme-based biodegradation. Additionally, research efforts may target the development of biodegradable alternatives to PET and the integration of enzyme-based biodegradation into existing waste management infrastructures. In this case, we have a combination of both. Or, as the professor said last time, we might come up with a solution and sell it for $1 billion.